In the name of God, my name is Samira Babaei. The subject of our discussion is divide and conquer approach to long genome sequence alignment. The contents include abstract, introduction, related work, problem definition, LCS-DAC implementation, evaluation and conclusion, and future work. Optimal alignment of DNA sequence is a reliable approach to discover mutation in one sequence in comparison to the other or to discover the differences between two sequences. Needleman-Wunsch and Smith-Waterman are the most applicable software for alignment of the sequences. Their performances are excellent with short sequences, but as the sequences become longer, their performance degeneration grows exponentially to the point that it is practically impossible to align to complete human DNAs. Alignment process is essential in diagnosis of genome-related disease. Many researches are done to find ways of performing the alignment with tolerable time and memory consumption. One effort is breaking the sequences into same number of parts and align corresponding parts together to produce the overall alignment. In this research, the method for breaking long sequence into smaller parts is based on the divide and conquer approach. The breaking points are selected along the longest common subsequence of the current sequences. The method is demonstrated to be very efficient with respect to both time and memory utilizations. Alignment of two sequences is the arranging of sequences such that similarities and differences are shown in the best possible way, while the order of elements of each sequence is preserved. There are a variety of purposes for alignment, such as revealing the relations between genes and proteins, understanding their homology and functionality, diagnosing disease, producing drugs. The optimal sequence alignment has the principle of optimality, and it is successfully solved by the dynamic programming approach. The two most utilized approaches, the Needleman-Wunsch and Smith-Waterman algorithms. The time complexity of Needleman-Wunsch and Smith-Waterman algorithm is OMN. Their problem is the time complexity and hence their high execution time for aligning line sequences. In addition, its space complexity is also high. One way to reduce the alignment execution time is to break the two sequences into many pairs of smaller sequences when possible and align each pair separately. The current research is in the same direction to higher an efficiency and the quality of globally aligning two genome sequences. It uses the longest common subsequence technology to find anchor points, divide and conquer approach to recursively break pairs of longer sequences, and Needleman-Wunsch algorithm to align every short pair of sequences. In this paper, a new divide and conquer approach to genome sequence alignment is proposed. The division is align the longest common subsequence of the two sequences, which is located approximately in the middle of the two sequences. Only a section in the middle of the two sequences are selected in which the LCS is thought. If the discovered LCS or longest common subsequence is not long enough, the section length is enlarged until a reasonable length LCS is obtained. The novelties of this approach is highlighted in the following. It is much faster than either of Needleman-Wunsch and Smith-Waterman algorithms. The longest common subsequence is removed from both sequences and hence from further processing, which further saved both execution time and memory space. It is faster than the state-of-art anchor-based method, which also used some kind of division. The space requirement of the method is extremely low. Human genome sequencing and analysis formally started in 1990, and with it, computational method, spatially alignment, becomes an important part of any genome analysis activity. Some aligners are Needleman-Wunsch, Smith-Waterman, BLAST, Mummer, GS-Align. Two genome sequences, S1 and S2, are inputs to the longest common subsequence divide and conquer approach. The output would be the global alignment of the two sequences in which differences and similarities of the sequences are clearly recognizable. It is assumed that the sequences are composed of characters A, C, G, and T. The length of the sequences could range from a couple of nucleotides up to the length of a whole human genome. In this system, we'll be able to align similar sequences of any sizes up to the length of a human genome. If both sequences are short, less than 100 base pairs is assumed here, the Needleman-Wunsch will directly be utilized. Otherwise, the dividing and conquer process will systematically break them into many pairs of short sequences. The gene sequences tested are BRCA1, ATF6, CFTR. These genes are taken from 1000 Genomes Project dataset. The bacteria sequences tested are E. coli, Shigella, and Salmonella. The computer used for the experiment is Intel and Core i7 and 6 GB RAM and Linux Ubuntu operation system. The results showed that on the average, LCS DAC 2.61 times faster than Mummer and 1.29 times faster than GS-Align. Another major area of comparison is the accuracy of the method. Results shows that the method presented in this paper is more accurate than others in all cases. The accuracy of LCS DAC is the highest. The accuracy of Mummer is 86% of LCS DAC and that of GS-Align is 66% of the LCS DAC. In this paper, we introduce LCS-DAC, which is a novel long DNA sequence aligner based on divide and conquer approach. The time complexity of the method is analyzed and it is shown to be superior to traditional aligner. The space complexity of the algorithm is also calculated to which is better than all classic aligner such as Needleman-Wunsch and Smith-Waterman method. Further, it outperforms any state-of-the-art method. The sequential version of the presented method is implemented, and it is compared against two state-of-the-art heuristic aligners called MUMER and GS-Align. It is shown that on the average, the proposed algorithm LCS-DAC is 2.61 times faster than MUMER and 1.29 times faster than GS-Align. For the accuracy, we show that the accuracy of MUMER is 86% of LCS-DAC and that of GS-Align is 66% of the LCS-DAC. Thanks for your attention.